Dear students, today I am with the sixth chapter of class 10th science, that is life processes. In my previous PPT, I have already discussed with the type of nutrition, giving emphasis to the autotropic mode of nutrition. Here I am just going to discuss about the holozoic mode of nutrition that takes place in the animals. Let us start from the definition of this holozoic mode of nutrition. Holozoic mode of nutrition, it is the nutrition which involves the 5 steps, that is ingestion, digestion, absorption, assimilation and egestion. So you can simply say this is a process in which the food is ingested, digested, absorbed and assimilated in the body of an organism to produce energy for its growth, development, maintenance and survival. What happens to the undigested food? It is being egested out or expelled out from the body. Starting from the amoeba, which is a unicellular organism, unicellular eukaryotic organism, it do not have any shape, so it takes its food by means of the phagocytosis. Phagocytosis means engulfing the whole solid particle in its body. How does it take food? As it senses the food, it just shoots out the pseudopodias from the two sides and it captures the food in the food vacuole. This food vacuole moves into the body of this amoeba within the cytoplasm, and then it is treated with the enzymes over there. The food is digested, then absorbed, and it is being assimilated or used up in the cell of the amoeba. And what happens to the undigested food? It comes in contact with the membrane and it is being given out. Now, the mode of nutrition in human beings. Human beings also shows the holozoic mode of nutrition. How does it happen? Human beings, they have a proper digestive system, and in this digestive system, you can see the pass and the passage of the movement of the food. The food is ingested to the mouth, and here through the mouth, from the mouth, it moves to the esophagus. Through the esophagus is a food pipe. Through the esophagus, it moves to the stomach by means of the peristaltic movements, or this is known as the peristalsis, and from stomach into the small intestine, large intestine, finally the rectum and the anus. Let us see the structure of our digestive system. You can see the complete digestive system, the way it is being placed in our body. The digestive system, that is the first part, is the mouth, or we call also, it leads to a cavity known as the buccal cavity. Also we call it the oral cavity. What do you see over there? You see the teeth, you see the tongue, a muscular structure, the tongue, as well as the saliva, which is being poured by 3 pairs of salivary glands that open into the mouth. These are called the salivary glands. Then after that, the food is properly masticated, and then the masticated food, it just travels down through the pharynx. In pharynx is the funnel shaped structure or the opening through which the food enters into the food pipe, or we call the esophagus. This is a tubular structure which is called the esophagus. From the esophagus, the food enters into our stomach. Then stomach is a sac where the digestive juices are being released, or we call it a gastric juice, and from here the stomach is connected to the small intestine. You can see a small intestine, it is a long coiled structure, measures near about 5.5 meters to 6 meters. So it remains coiled in our body. Then the food from the small intestine enters into the large intestine. Remember, in the small intestine, the complete breakdown of the food as well as the absorption of the nutrients takes place. The food enters in the undigested food, you can say, it enters into the large intestine, which is slightly wider, and thus it remains as in the body as it is being shown in this diagram. The large intestine is the place where no digestion of the food occurs. This is the place only where the water absorption occurs. This question is generally being asked in the board examination. This large intestine, it is being divided into certain limbs. You can normally see this one is the cecum, where you can see a small comma shaped structure which is called the appendix, a part of this cecum. Cecum, we don't have the cecum, in fact it is too much reduced to the appendix also, you can say, because we have changed our food habits from the herbivores to the omnivores, or to the omnivores. So So they are the cecum is the place where large number of bacteria which help in the digestion of the cellulose, they are generally found. So we don't require it, so it remains as a vestigial organ. It just helps, throws light on evolution. Then you can see the large intestine, the limb, the upper part. So you can say cecum, colon, as well as the rectum. These are the parts of our large intestine. In the last instance, I already mentioned that the absorption of the water occurs. Rectum is the part of the large intestine where the waste is being stored, and through the anus it is being expelled out. Now, again, human digestive system, you can just see here I have just shown you the diagram of the buccal cavity. See how the teeth are being arranged, and the tongue, you can see the palates in this buccal cavity. This is the structure where I've just shown you the glands which are associated with this digestive system. I've told you in the beginning that in the buccal cavity we have the three pairs of salivary glands which pour the saliva in the mouth. Now the largest gland of the body is the liver. Liver, it form it it forms the bile juice, and this bile juice is being stored in the gallbladder. The second largest gland of the body is the pancreas, and this pancreas, it is also known as the mixed gland, or we also call it a compound gland, because it releases the pancreatic juice as well as it is also involved in the releasing of hormones. That's why it is known as the compound gland or the complex gland or mixed gland also. So the digestive juice or this pancreatic juice is being released. Here you can see just below the stomach a C-shaped tube here. This is called the duodenum, the first part of our small intestine, which we call the duodenum. The whole structure is that this whole part is the duodenum. This internal part, as the most dissected part, has been shown to you. This is the reason where the bile juice and the pancreatic juice and the walls of the intestine releases the juice, and the further digestion of the food, complete digestion of the food, occurs over here. Then the food enters into the remaining part of the small intestine, then large intestine. Let us see now the mechanism of digestion of the food. Starting from the buccal cavity, see we have teeth, so we should be aware about the dental formula. The dental formula is 2, 1, 2, 3 pairs in each jaw. This 2 means the incisor. We have 4 types of teeth. The incisors, so it means 2 pairs of incisors, 1 pair of canines, 2 pairs of premolars, and 3 pairs of molars in each jaw. So it means 32 teeth. Then tongue. Tongue is the muscular structure which senses the taste and help in rolling of the food in the mouth. Saliva. This saliva which is released by the salivary glands, it lubricates and digests the starch in the mouth. This consists of, what is the composition of the saliva? It consists of water, salivary amylase. This is a very important enzyme which helps in the breaking down of the starch to maltose, lysozyme which helps in killing the bacteria, and the mucin, that is the mucus, which helps in lubricating the food. In the stomach. Now in the mouth, the food is completely masticated. It turns into a ball-shaped structure which is called the bolus. This bolus moves through the food pipe into the stomach by means of peristaltic movement. Once the food enters the stomach, you see the gastric juice released by the gastric glands present in the inner wall of the stomach. They act on the food. What is the composition now of the gastric juice? Composition means what does it contain? It consists of mucin, again the mucus, hydrochloric acid, as well as the protein digesting enzyme known as the pepsin. Remember, this protein digesting enzyme is not released in active form. Suppose if it is released in active form, what will happen? It will start acting on the protein or on the walls of the stomach and start digesting it. So as soon as it comes in the contact with the HCl, then only this pepsin in active form is being converted. So you can say the inactive form of pepsin which is being released in the digestive juice is the pepsinogen. So HCl convert pepsinogen into pepsin. Next part is the small intestine. Here in the stomach, the food is completely masticated, or you cannot masticate it, but better you can use, it is being churned. and convert into semi-solid paste. Only protein digestion of the protein occurs over there in the acidic medium. So the food is known as the chyme, chyme, chyme. When the food enters the small intestine, remember here in the small intestine, I've already told you the bile juice from the liver is added to the food, pancreatic juice from the pancreas, as well as intestinal juice, which is also known as rich in succus entericus. It also get mixed with the food. Here I have just made the flow chart showing the complete digestion of the food. You can just learn it also. Here in the mouth, you can see the salivary glands. We secrete the saliva. What do they do? They convert starch into sugar. And this sugar is known as the maltose. Then the food enters into the esophagus only by peristaltic movements. It just moves to the stomach. In the stomach, the gastric juices, they contain the pepsin. Pepsin, I have already told you that this is released in inactive form. That inactive form is known as the pepsinogen. So what is the role of pepsin? It breaks down the proteins into smaller molecules. And HCl, it makes the medium acidic. Whereas mucus, it protects the inner lining of the stomach also. Then come to the from stomach, the food is entering into the small intestine. In the small intestine, liver is releasing the bile juice. This bile juice, especially here, I just want to mention bile juice. It consists two things. Number one, bile salts, and second, bile salts and bile pigments. Bile salts. Bile salts, they are only the one which helps in the digestion. Bile juice do not contain any enzyme for the digestion of the food. So bile salts, first of all, they neutralizes the acid that enters with the food in the small intestine. And second, a function of the bile salts is they help in the emulsification. I'm using a new term known as the emulsification. Emulsification of the fats, in which the big fat drops, they are being broken down into very small fine droplets so that they can be easily digested into their components. We call it emulsification. About bile pigments, these are the pigments which are formed, yellow pigment, yellow and green pigment, which are formed by the breakdown of the hemoglobin in the liver. So they don't have any work in our body. What happens? These are being expelled out through our feces. If somebody asks you what is the main role of it, you simply say that they impart color to our feces. After that, the pancreas releases the pancreatic juice, and intestinal wall releases the intestinal juice, which is rich in the intestinal enzymes, which help in the breaking down of carbohydrates, fats, as well as protein. When the carbohydrates are being broken down, they break into glucose, fats. They are made up of fatty acid and glycerol. So they are broken down into fatty acids as well as glycerol. Proteins, they are being broken down to amino acids. Finally, now pancreatic juice, if I say, pancreatic juice consists of, here it is being shown only two main enzymes. Here I would like to add one more thing, that is the amylase also. Starch digesting enzyme. Please note it down, though in the flowchart it is not there. That is pancreatic amylase. Here you can say amylase, you can directly pancreatic amylase. This pancreatic amylase, the term refers to the starch, amylose word. So pancreatic amylase means the enzyme which helps in the digestion of starch in the intestine. We call, and it is released from the pancreas. So name has been given pancreatic amylase. See here the trypsin. Pancreatic juice contain, I've told you, pancreatic amylase, which I'm adding out over here. Then the trypsin as well as the lipase. Trypsin again is released in inactive form, trypsinogen. The active form is known as the trypsin. What does it do? It acts on the proteins in basic medium. Remember, the enzymes, those which are working in the small intestine, they work in basic medium. So trypsin, it will break the protein, all the whole protein will not be broken down in the stomach. It will further break down the proteins into smaller molecules known as the peptones. And lipase, see, lipase, it converts the fats, emulsified fats. I have already mentioned, I've told you about the emulsion. So emulsified, the very very small fine particles of the fats here, they are being broken to lipase help in the breaking down the emulsified fats into fatty acids and glycerol. All these composed, and then finally they are being absorbed by the body. In the last, here you can mention, see, large fat globules, they are converted into small fat globules with the help of the bile juice. This bile juice, what is there? That the bile salts. So this process is known as the emulsification. Question can be asked, what is emulsification? Here complete digestion of the fats of the food has been taken place. The digested food now it moves into the food is being absorbed by the intestinal villi, or you can say villi. These are the finger-like projections which increases the surface area for the absorption of the food. Finally, the food enters into the large intestine. Here, no digestion of the food occurs, no absorption of the nutrients occur, only the absorption of water takes place. From the large intestine, rectum is the last part of the large intestine. Here, the waste is being temporarily stored in it, and which is being ejected out or egested out periodically through the anus. This is all for today, and we'll see you in the next PPT with the next topic. Thank you very much.